Okay, so we move to what we did on cerebral blood flows and the tools we have developed, and one of the tool actually, which is more than a tool because it's a set of tools, actually a platform, can be used also for the eye. And then Lorenzo will present the actual application and maybe do some demonstration about what we have at the moment. Okay, so the agenda is that we have developed some work from the mathematical modeling point of view regarding the cerebral blood flow, and in particular in the Venous network. In the Venous network, there is less, literature or knowledge on the blood flow. The shape, for example, of the vessels is different. The way the blood is driven through the vessels is also different. Regarding the modeling, what we should use as a constitutive law, this is also not so clear. With respect also to boundary conditions or setup of our possible models, this is also complicated. And if you look at the literature, you can have a huge variability in terms of parameter sets, for example, for the Venous network. And then the toolboxes that we have developed, in particular in fluid dynamics, which are available. This is actually the fluid toolbox, which is in the Docker image that we deploy. This is exactly the same tool. It was validated for rigid and compliant vessels, benchmarks that we did. So this will be the second point. And the last point, this is for us a very important tool that we have developed. It's called AngioTK. This is a platform to be able to reconstruct vessels from medical images and actually from various types of data. I can maybe point out some applications that we have here where we actually do reconstruction, but not from medical data. Actually here, what you see here, I don't know if you see the pointer, is that is actually an electrical equivalent model for the eye and the brain system. So at the top you have the brain part, the compartments regarding the brain. Here you have the lamina cribrosa. Here you have the eye, you have the eye. So you have a set of electric components that are plugged together. Actually, we use Open Modelica in order to do this kind of model. This is extremely simple in Open Modelica to do that. And then we can run through Open Modelica this system. This is a systemic view of the model. And then here, our interest is actually to be able to make zooms into the system and have 3D models replacing some compartments of our 0D model. Okay? To this end, we have made various developments. So I start with the Venous network part. In the Venous network part, as I said earlier, there is less, in terms of modeling, there is far less things which are known on this system. And so with my colleagues, we actually did a rather intensive study using deterministic analysis tool to do a sensitivity analysis with respect to various points, boundary conditions, inflow, outflow, and also constitutive law, how they impact on the modeling with respect to measurements that we have. Is it, like, the question is whether the conditions are very sensitive or not, or the constitutive law choice is sensitive or not. I mean, does it impact in an important way the flow? To do this study, actually, the actual processing for the sensitivity analysis actually costs more. It costs more than the actual computation because we had to compute a lot of. We had to do a lot of computation, which required more compute power than the actual computation of the blood flow in order to extract this information. So this is available in some publication if you are interested in, and this allows us to provide some first steps toward the understanding of the blood flow inside the cerebral venous network. So these tools are integrated also in our in what we provide in our toolboxes. Another point, important point, is that in the brain, in the venous network, actually the blood is driven by pressure. This is you have a pressure drop between the output of the capillaries to the thorax. You have a pressure drop, which drives the blood flow. And here, in order to take into account pressure in a blood flow model, for example, say Navier-Stokes or Navier-Stokes, if you want to do that, you would need like non-standard boundary conditions. And so this is a recent work which has been published in CMAME, where we did the theoretical as well as the numerical assessment of this boundary condition. Here we can impose pressure, but in order to impose pressure, you need also to complement this condition to also control the tangential velocity components. They has to be zeros. So for example, here, if you at the extremities, so if you now zoom at the blood, here you see the pressure drop. And if you look at the velocity, you see at the extremities either at the inputs, so here the blood comes from the capillaries, or at the output from the jugulars, you see that the velocity is really straight out of the, or inside. And it models also nicely the fact that the tube, we cut the tube, but the tube continues, right? So if you use standard outlet boundary condition, you will get like, you know, when you are a wrong outflow condition typically. So we did this if you are interested, and it's also, of course, delivered with CFD toolbox that we provide here. In terms of now, in terms of benchmarking, so we benchmark the CFD code that we use for the blood flow. First, on the rigid vessel. Actually, it's not really rigid vessel here. It's actually an FDA. FDA is Food and Drug Administration. So they actually, it's a device which is here, which allows to combine various effects that you see in medical devices, that you will have accelerating part, decelerating parts, recirculating flows. And that's what you see in real medical devices. A lot of data is available online. Also different codes, software, but also with PIV measurements which are available. So it's also published. This is a nice way to benchmark. Ah, someone is connected. Okay. Another benchmark is with respect to compliant vessels. So in that case, this is an experiment we did in Strasbourg. So we put a stent, well, not a stent actually, sorry, something that could be used for after surgery to replace a part of an artery. So it's actually here. And you have a bifurcation, and then the bifurcation comes back. So you see the simulation here. So it's free structure interaction. And this is here. The flow goes in this direction, say. And it's fluid which has similar viscosity as the blood. And we put this system into an MRI, and we acquired the MRI data. But we control completely the flow. So we know exactly the setup of the flow inside the system. So what we did is that we managed to reconstruct the entire geometry from MRI measurements that we did. And we simulate the blood flow, of course, in the true and the reconstructed geometry. And we can compare with the acquired data. So it's an excellent benchmark in order to verify that the tools that we build are making sense and they do a good job. So this work, now, of course, okay. So this work has been, in particular, has been put into the platform which is called AngioTK. Sorry, I have someone up. It's called AngioTK, and it is developed within a project called Eye2brain in France. And this will be linked to the Eye2brain project. So we take MRI images of the brain. It takes some time. I was part of the people who went into the MRI. And then from the MRI images, you apply filtering and segmentation tools. Then you reconstruct computational meshes, you do the blood flow, and then you using particle tracing and MRI modeling, the block equations, then you can reconstruct virtual images and see whether you can reconstruct images that are similar to the actual images that you got from the real MRI. And so that's what we actually did here on this model that we control completely to make sure that we are doing things right. So in this AngioTK platform, it's actually a tool chain, so it's complex tool set. So we have ITK from Kitware for the angiographic image analysis, Fil++ for the blood flow, VMTK, VTK for image generation processing. Also for path opening, this is more like filtering technique. We use another software, Gmesh for mesh generation, and GEMRIS for the MRI simulator. So you have quite a bit of steps into the application. And also interesting is that we have quite a bit of data which is stored in currently in a MIDAS server. It's a kind of well, if you like, database and nice interface to deal with data developed by Kitware. So we have quite a bit of information stored there. It would be quite interesting to connect CCAN to MIDAS in particular. And using this data, we have about 40 patients among myself. And with this data, the data will follow a journey towards mathematical algorithms. So it starts top left. We have 3D images. So the MRI data is actually a stack of 2D images. So you have about this part of your body in our data. And then it follows the path here, which I'm going to present. So the first step is that here we want to emphasize the tubular structures inside the image. This is a filtered image where the algorithm actually emphasizes the vessels, because otherwise it will be like inside quite a bit of noise and other kind of information. So in order to be able to extract this information, you need first to filter the image in a clever way. And in particular, we use this HORPO filtering tool, which allows to recognize inside images tubular structures. So here this is before using the tool, and here this is after using the tool. So you see that now it's much easier to do some kind of extraction. So here it is similar on different kind of, on a different image. This was here a 3D image, And now here it's a 2D image, and you see that we have emphasis each time the tubular structure. Another feature of this filtering technique is that while detecting the tubular structure, it allows also to give an estimation of the orientation of the structure. So here an example of a 3D image before using the tool and a 3D image after using the tool. See? So this is extremely important for what follows, because if you don't have that, then you get a lot of garbage. And here you see, even though we have nice, seems like nice data, if you zoom, you realize that it's not so nice actually. Here is an extraction. So we do a segmentation. We take thresholds inside the image, and then we just extract. You can do that with ParaView if you like. But you see here, like it seems that there are some parts which are not connected. There are some weird stuff. You look here. Well, you can. If you look close, then you will see that there might be some not so nice features, and in particular, it's not quite useful or usable by a flow simulator. So what we do is that from this data, we build so-called center lines. So we manage to build a kind of tree of curve or set of curves inside the vessels. So you see these curves here. But not only do we have these curves, but we have also an information about the kind of local diameter of the objects. Okay? And from this information, now we have these curves, we have these diameters, local diameters. So we have a set of points and connections between those points, plus diameter information. From this, we are going to reconstruct a 3D image. So we go back to imaging, basically, but only with the center line information and the diameter. We have also a clever algorithm in order to avoid, we build so-called distance functions to distance functions. So at a point here inside the image, we will know at which distance we are to the boundary of the vessel. Okay? So at each point, so when it's blue, it means that we are inside the vessel. When it's red, we are outside the vessel. And using the 3D image, so here it's a data structure that we used to localize very, very efficiently the vessels, to do the segmentation afterwards in order to evaluate the distance function that we build in an efficient fashion. So once we have done this, we extract the level 0 of the distance function, and the level 0 is actually a level set which provides you the boundary, the lumen, that is the interior boundary of the vessels, the vein or artery. And at this point, we have a nice geometry, not yet usable. Here, the next step is actually we need, if we make, I have some video with some zooms later, but here at the extremities, the extremities are not dealt with. The surface of the mesh can be quite fine, for example, or too fine. So maybe we want to de-refine or to further refine. It depends on what we want to do. So here we will fix all the extremities. We will tag all the extremities. Eventually we will be able to create the arterial wall also, if we want to do fluid structure interaction. And we can do the mesh for the volume, and we have, of course, the partitioning of the mesh. Yeah. So, for example, here I do a zoom of the previous. You can see we have a nice mesh here. And then we launch the toolbox in order to have blood flow inside the system, with different kind of models available, different kind of boundary condition, different kind. We have some efficient solvers for Navier-Stokes and free structure interaction available. We deployed on Prace infrastructure those solvers on hundreds and thousands of cores. Yeah. And we can also, what we do also is in situ. We have in situ facilities to monitor the simulation through ParaView. So there is the network is actually quite slow, so it's a pity. But here there is a video. So this is a 3D image. Here, the first segmentation with quite a bit of garbage. Then we extract the center line. We build the sign distance function. We do the next extraction with remeshing to get some nice geometry. And then here, for example, you can see the journey the data makes. So this is the first extraction. This is not good. This is not good here from MRI data. For our solvers, that would not be good. And then we do the reconstruction, we do the remeshing. So here you see that we managed to de-refine the mesh. Then we can open the mesh and do properly boundary conditions. So you can see here, this is how the extremities look like when we do the extractions, you know, like. But what we need to do is to open the extremities and then do a nice cut into the system to impose the boundary conditions. If you are wondering, this is all done completely automatically. There is only one step which is not automatic, which is the center line computation. This is still a challenge. This is an open problem, and various teams around the world are actually looking at this. So this is for the boundary conditions. I'm not going to bother you with that. So where are we with respect to the status of this platform? So AngioTK is part of Eye2brain. This is one of the many tools that we actually use here. So it's developed within GitHub. We have a Docker image which is currently built, and it is built automatically using a continuous integration tool which is called BuildKite. This is not open, but this is absolutely awesome tool because you can use your own compute facility or compile farm, whereas with other tools like Travis you can't. And all these tools require quite a bit of time, you know, to set up, compile, and everything. So being able to plug your own compile facilities is extremely interesting here, and we can deliver the Docker images to our partners. We have this data server where we can actually take the data. I put in parentheses a link with CCAN because this is not existent, but actually I would be thrilled to test and make sure that it works well with CCAN. And finally, we have kind of continuous integration test of all this data. We have this big data set, and whenever we make a test, a change in the code, in our algorithms, then we can run through all the data and verify that it's still going properly through the data, and we have all the history, so we can see from different changes in algorithm if we are still, if we fix the bug, or if we introduce a new bug because some data set is not working anymore, or things like that. So we have set up something like Travis or BuildKite on the data set directly to verify that our algorithms are running smoothly. So AngioTK is an important asset for Eye2brain. So this is the kind of thing that we do. Like, you know, you have the Venous network here, you have the arterial network here. You will notice for the people who are interested in you had the flow here. Here is geometry. That the person here doesn't have a cycle of Willis. You know, you have these two sets, two branches, like the blue and two parts here, the blue and the red. And here they are, a lot of people have a so-called cycle of Willis, which connects those two parts. And the cycle, you may not have it. And that's a problem if you have a stroke, for example. If you have, yes, exactly. An aneurysm, or if you, then because it will, the cycle of Willis will actually, could help with redirecting some part of the blood flow, right? So, but there are still some work to do, in particular with respect to the center line part. So all these tools, the toolbox and everything is actually available, and we need at some point to plug this on the Eye2brain part, which is not yet the case. We actually can do, we are also looking in the direction of, we start to, as Alan Harris showed you, we start to have angiographies also for the eye. So the idea is that from this, we discussed with a company called OptoView that can provide those angiographies. And the idea would be also to use AngioTK to reconstruct local part of the tissues with the vessels and using AngioTK. So now I will pass the microphone to Lorenzo, and Lorenzo will show you the actual application that we have developed and maybe make some demonstration.